Hi everyone, this is Andy and welcome back to another educational video from Med School EU. And in today's lecture, we're going to take a look at the cell division, but more specifically, we'll look at the sexual cell division in the means of sexual reproduction through the process of meiosis, and as well, we're going to define some terms, some confusing terms in chromosomes. Alright, so to make this video a little bit less confusing, because we are going to be talking about a lot in terms of chromosomes and their structures and how they're defined, so let's go over some chromosome terminology first so that we know what's going on when we're looking at the process of meiosis. So first of all, if we're looking at a cell from this sort of view in the microscope and we see that the chromosomes are in this sort of loose arrangement, you can't really tell which one is which, and they're all kind of in this sort of convoluted structure. This structure we would call, let's label this as chromatin. This is not chromosome. These are chromosomes. However, if we're looking at a structure that is loose like this, if the chromosomes are looking loose, this will be called a chromatin. And another definition of chromatin, because it is simply defined as, it is going to be defined as DNA, so the genetic material, plus its proteins. So it would be in this sort of convoluted shape. It would be raveled and packaged in this sort of way. Now these proteins here would be histones. They would be histones, and the DNA all that is connecting to the histones and winds around, this would be called linker DNA, as it links each of the histones together. And that's just how we can have, meters of DNA just raveled in this sort of structure called a chromatin. So this entire thing here would also be called the chromatin. However, if we are looking into division, so even in the process of mitosis, you're going to see chromosomes that would be lined up in this sort of arrangement, and they would no longer be called specifically chromatin, because a lot of times they would be referred to as, let's label this here, this would be just one chromosome. All right, so that's the chromosome. Chromatin is this structure right here, so DNA and its proteins. However, there's more to it. So if we're looking at the chromosome in a closer view, there's another structure that we must define, because this is going to be called a homologous pair, because as you can see, it has two. And both of these would be called together, they would be called homologous pair. Why is it homologous? Because they code for the same genes. So let's say this would be from the father, we'll label that as male, and this would be from the mother, we'll label that as female. And they code for the same genes. Mother and father, they would have the same genes on each of their chromosomes. However, each of these genes, they would have different versions of them and different versions of genes. So let's say we have a gene right here, and the father has a gene right here on the same location, but they're different versions. They might be, this one might be heterozygous for brown hair, and this one is homozygous for brown hair. And the expression, so the organism could only express one of these alleles. So let's label these as alleles. Alleles are simply versions of genes. Okay, So now we know chromosome, that is the chromosome structure. We now know chromatin. There's another thing that we should note, and that would be called chromatid. So each of these, from the homologous pair, so the homologous pair is made up of two sister, so this would be labeled as sister chromatid. So each one of these is a chromatid. And two sister chromatids make up a homologous pair that is a chromosome. So that's how we would label most of the terminology in chromosomes. And another thing that I must point out, because it does get confusing a lot with students, is that we do not define chromosomes by the number of homologous pairs. So you could do that. However, it gets confusing once these pairs have split up and we still have the same number of chromosomes. How come is that? Well, because we designate the number of chromosomes to the number of centromeres, number of centromeres. That's how we count the number of chromosomes. So here we have this chromosome is connected by one centromere. So centromere is just the region of DNA in the middle. So that's the centromere, the region of DNA in the middle of the chromosome. So two homologous pairs are linked up together by this one centromere structure. Now, the number of centromeres that we have equals the number of chromosomes that we have. So we may not have a homologous pair. We would have a sister chromatid. So once these are split up, each sister chromatid would have its own centromere. And therefore, once these are split up, there would be two different chromosomes. They're homologous pairs. However, if they're split up to different sides of the cell, as they do in mitosis, they would become two chromosomes instead of just one, because now we have two different centromeres. We're counting by the number of centromeres. That's very important, so you do not get confused in the process of meiosis and the process of mitosis when you're counting the number of chromosomes. Now, another thing to point out, if we have a chromosome like this right here, there's another confusion and misconception about what a centromere is and what a kinetochore is. So we talked about kinetochores in our last lecture about mitosis. So kinetochores would be structures like right here. Let's label this as kinetochore. And kinetochores are not DNA. They are proteins. They're assembled proteins that are assembled during mitosis and also during meiosis 2. They are assembled on the protein, on the DNA, specifically on the centromere region of DNA, in order to attach microtubules. So microtubules would assemble on them, and therefore the microtubules can pull them apart and break the centromere and the homologous pair. They would break them up into two different chromosomes, into their sister chromatids, and the kinetochores would be acting in that manner. However, kinetochores are not centromere. Centromere is DNA region. DNA region. It's simply the central region in the DNA. And kinetochores are assembled proteins that assemble on the centromere in order to split the homologous pair into its sister chromatids during mitosis and meiosis 2, as we're about to see. All right, so moving on into the process of genetic recombination in eukaryotes. Specifically, we'll look at eukaryotes as humans are eukaryotes, right? So we will look at the process of genetic recombination because we we all reproduce through sexual reproduction, not asexual reproduction. So let's define the two terms and let's figure out which cells actually go through the process of asexual reproduction and the process of sexual reproduction. Asexual and sexual reproduction. because we have some cells that go through asexual and some cells that go through sexual reproduction. So let's figure those out. First of all, sexual reproduction depends on the process of meiosis. So first of all, we're going to take a look at the life cycle of eukaryotic cells in order to figure out which cells actually go through asexual reproduction and which ones go through sexual reproduction. So here, let's label some of these things here. So we have our animal, right? So that would be us, human. Let's label this animal, since we're looking at eukaryotes in general. Here we have a zygote. And right here we have gametes. And gametes are, so that's the egg, the female egg, and that's the male sperm. They're called gametes. They're our sexual cells that would go through the process of here fertilization. And once they have fertilized, so once the sperm has fertilized the egg, they would form a zygote. And the zygote would obviously go through many, many stages of mitosis in order to become a full-grown organism. However, let's define which one's asexual, which one's sexual, and how the life cycles work and what they're called. First of all, the longest stage, so the animal life cycles are called diploid life cycles. So let's label this entire thing as diploid phase. And why do we call it diploid? Diploid meaning that it's 2n. So it's n is the number of chromosomes in a gamete multiplied by 2. So as we know, humans have 46 chromosomes, or they have 23 pairs. So each of the gametes would have 23 chromosomes, not pairs, just 23 chromosomes. So here we got 23, here we got 23. Once they combine in the process of fertilization, they form a zygote. And the zygote right here, so once they have fertilized, they become diploid because it's multiplied by 2. 23 becomes 46. So 46 chromosomes, and we have 46 chromosomes during our entire life cycle. That's why we're called diploid phase, because we exist as 46 chromosomes. Every cell that you look at that is not undergoing mitosis, it would have 46 chromosomes the entire time. However, we do have a stage called haploid phase, and that's only when we're going through meiosis. So let's look at the next stage here. Once the animal is fully grown and is able to reproduce and form viable sperm cells and viable gametes, these cells, before they become gametes, they're called germ cells, and they go through the process of meiosis in order to become gametes, which in turn would fertilize, would go through the process of fertilization and become a zygote and form another animal. However, this stage is going to be called haploid phase. And why is it called haploid phase? Because only 23 chromosomes. So if we are the male and we have made sperm cells through the process of meiosis, the sperm cells are going to have n number of chromosomes, which is 23, only half the number that we would normally have during our life cycle. Only the gametes would have 23 number of chromosomes, and that while the organism, I guess, exists in just this one cell of a sperm or an egg, they would only have 23 chromosomes, and therefore that is called haploid phase because it's half of what we would normally have. Once we go through fertilization, of course, we go into the diploid phase, and the germ cells would divide again in order to form more gametes, and the cycle continues on and on. So let's really discover which cells go through the process of mitosis. So asexual reproduction, let's label it here, mitosis. We have mitosis, which would be asexual reproduction. And which cells go through asexual reproduction? All cells go through asexual reproduction except germ cells. And all the cells in our body, so every single cell that you can think of except for our reproductive cells, that would be our sperm and the egg, that would be the gametes, all the other cells go through the process of mitosis that we already know, and all of these cells would be called somatic cells. They go through the general process of asexual reproduction of mitosis. Now, if we are going in the stage of haploid, so which cells go through sexual reproduction? Because sexual reproduction involves fertilization and the formation of a zygote. So if we're looking at the other side, we have meiosis. Right? Meiosis would go through sexual reproduction. That's what depicts the name. And meiosis, the cells that go through meiosis would be called germ cells. Germ cells that would form gametes. And gametes would be either the sperm or the egg. Now the gametes would then go through fertilization, and once that happens and the zygote is formed, we go through the process of mitosis over and over and over and over again until the animal is fully grown. Another way to really figure out why we say diploid and haploid. So in mitosis, if we have a cell, here we have a cell that, let's say, has, let's make this a different color. It has 4 chromosomes like this. 4 homologous pairs of chromosomes. Now, these would be 2N. They would be diploid. Now, once they go through the process of mitosis, right, so we can label here mitosis happens, asexual reproduction. And what this forms is, of course, 2 cells that would have 1, 2, 3, 4. 1, 2, 3, 4. Now notice, this is also 2N, and this is 2N. We have not reduced the number of chromosomes. Why? Because we have 1 centromere, 2 centromere, 3, 4 centromeres. Here we have 1, 2, 3, 4. 1, 2, 3, 4. We still have 4 chromosomes just as we started from. And that's why I say it's very important to know that the number of chromosomes is counted by the number of centromeres and not by the number of homologous pairs. Here we don't have homologous pairs. Each homologous pair has split into its sister chromatids. However, there are 4, and each sister chromatid has a centromere. Now, we count the number of centromeres. We're still at the diploid phase. This does not happen in meiosis. Meiosis goes through 2 stages, so there's meiosis 1 and meiosis 2. And we're going to take a look at the specifics of how that occurs, and then we can compare back into how that is related to the process of mitosis. Now, before we look at the specifics of meiosis 1, I wanted to go through just a general overview of what happens to the chromosomes so we're not confused about the haploid and the diploid phases. So, first, let's say we have a cell that has 2 chromosomes. We will have the red chromosome and we will have the white chromosome right here. So these are homologous pairs, and they they exist in the cell. Now, this would be a germ cell, so of course germ cell, because only germ cells go through meiosis. And now if we are going into the next stage, what happens is during the S phase, there's replication, right? So let's draw the same cell. And each of the chromosomes went through replication. Now, we have still 2 chromosomes because centromeres, 1, 2, 1, 2. We did not increase the number of chromosomes. We simply replicated the same one, but they're attached to the same centromere. Therefore, they are just simply sister chromatids that have formed homologous pair. So next, after the chromosomes have replicated, we go through the process of Meiosis One, and that's the one we're going to look at next in detail. However, what occurs with Meiosis One is that we end up with 2 cells now. Of course, the cells have split, and now we have the red on this side and the white on this side. Now, they also go through something called genetic recombination, and we will talk about that in detail. However, for the purposes of this slide, we're just going to make sure that we know what's going on with the haploid and the diploid. Now, after Meiosis One occurs and we have these 2 cells, each of these cells go through the process of Meiosis Two. So that's Meiosis Two. And what happens in Meiosis Two is we end up with 1, 2, and 1, and 2. So what occurs here is we have just the sister chromatid and the white one, sister chromatid. Right? So now if we count, let's go back and count the number of chromosomes we have, and looking at their haploid and the diploid phase. So here we have 2 chromosomes. So we are at diploid phase, 2N. Here still 2 chromosomes, diploid phase. Now here we have split up from Meiosis One, and we just have the single chromosome. Although it's a homologous pair, it's still just 1 chromosome. So here we enter into the haploid stage, which would just be N. So this is haploid. After Meiosis One, each cell becomes haploid. Now these cells would divide further, and they would form sister chromatids. However, it still has just the 1 chromosome. So this is N, 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 and N. And these are going to be gametes, either sperm or the egg, depending on male or female. And these gametes would then go through the process of fertilization with the opposite sex, forming a diploid. Okay, so that's how we would go through just the general process of meiosis to figure out what's going on with the chromosomes, so you know before we go into the specifics with Meiosis One. All right, so let's take a look at Meiosis One and what's going on in terms of the cell. First, we begin with prophase, as we do in mitosis. So the stages are exactly the same. We would have them as IMAT. Prophase, metaphase, anaphase, telophase. And meiosis would have quite a distinctive prophase because I would divide it into 3 different sections just to highlight 3 different things that are happening. So at the beginning of prophase 1, the chromosomes begin to condense into thread-like structures. So over here, they begin to condense, as we can see. And each consists of 2 sister chromatids as premeiotic, as a result of DNA replication during the interphase stage. And the chromosomes of 2 homologous pairs, we'll have the one long here and 1 short. And that's what I'm showing on the diagram. Now, another thing I wanted to clear up in terms of the concepts is that the structures right here, this all together, so both of these, all of this, if you highlight all of that, so these yellow structures right here, they are called centrosome. Centrosome. However, each individual structure, so if I'm highlighting this here, each individual structure would be called a centriole. Centriole. So do not confuse centrosome, centromere, and centriole. Three different things. Centromere is the region in the middle of the DNA of each chromosome. Then the centrosome are both centrioles. The structure is called centrosome, and each centrosome has two centrioles. So this stage we would have more specifically condensation. Condensation of chromosomes. That's the highlighted stage that we have, the first stage of prophase. So I'm going to have that as 1. Now next, moving on to the next structure of prophase, the second one is called synapse. So the lining up of each of the homologous chromosomes, that's called synapse. So I'm going to label this as synapse. And the next part of prophase is going to be recombination. So this whole process is called number 3 here, and recombination. So recombination is going to be the exchange of genetic material or chromosome segments between the homologous pairs. So once they have lined up, as you can see, the pink and the bluish one, once they line up, they do this crossover. So sometimes this is also called a crossover. Crossover. They go through a process called recombination. And recombination, simply exchange of genetic material and exchange of chromosome segments between the homologous pairs. As you can see what happens here, the pink then takes on this bluish, and the bluish takes on the pink. Now this all 3 stages right here, so all 3 of these stages, they would be called prophase 1. Why is it 1? Because of Meiosis One. So prophase 1. Next, moving on, right after prophase, we have an intermediate stage between prophase and metaphase, and this one is going to be called prometaphase. Let's label it here, prometaphase 1. Now the notable thing that happens here is that first of all, the nuclear membrane is gone. The nuclear membrane has disintegrated, and that marks the beginning of metaphase. Now prometaphase is the stage where the spindle moves into the former nuclear area, so where the nucleus was, and the spindle moves in there. And the kinetochore microtubules connect to the chromosomes. So remember we talked about kinetochores are assembled proteins on the centromeres, and they attach microtubules. So these yellow projections are microtubules. They come off each of the centrioles, and they would attach to the kinetochores of each chromosome. Now the chromosomes also start lining up in the middle of the cell, which would be called metaphase plate, as you will see in the next one. So here we have the real stage, the process of metaphase 1. And Metaphase 1, notably what happens is these chromosomes, they form the process, what is called, here I'm going to label this like that, and this is called a tetrad. So each chromosome is lined up on its side of the cell, and it's lined up in this dashed line is called the metaphase plate. And what happens is they form the tetrad. So two homologous pairs form a tetrad, and they line up in this sort of manner in Metaphase 1. Moving on to the next stage is going to be Anaphase following Metaphase. So let's label it Anaphase. Let's get the colors right. That's going to be Anaphase. Anaphase 1 in particular. And what occurs in Anaphase 1 is that the tetrad is broken apart. They're no longer lined up in this manner. And the microtubules have pulled each of the chromosomes to its side of the cell. Now also, the microtubules are going to begin splitting the cytoplasm and increasing the cell in this manner and making it elongated, because the microtubules are simply pushing each other apart from the cell plate. And this all occurs in Anaphase 1. Now moving on to the final stage of Meiosis 1, and this one would be called Telophase. Telophase 1. And what we have is the formation, so the narrowing, or what we call furrowing, as we did in mitosis. It would be the furrowing of the cell, and the cell would almost split up. And what we have, the result, is each chromosome, each homologous pair has now gone through the process of recombination or crossover. And this is where we get genetic variation, because now the chromosome that will be passed on, and this happens to all 23 chromosomes that would be passed on, each one of them would have some sort of recombination or crossover with the other one, with its homologous pair. And this is where we get so much genetic variation, and why we're kind of a combination between our father, our biological father, and our biological mother. And what we have is the bluish area is now part of the pink chromosome, and this little pink area is part of, this pink segment is part of the bluish chromosome. And that is the final stage in Telophase 1, and that's what we end up with. However, the recombination occurs in Prophase 1. That's the stage where recombination occurs at the very end of it. So moving on to Meiosis 2, and what we have, this is what we left off with. Obviously the furrowing finished with the process of interkinesis. So interkinesis would divide the cytoplasm. However, it does not, the germ cell that has undergone Meiosis 1, it does not enter into another interphase stage where it goes through a replication. It doesn't do that. There is no replication. That's important to know, because the chromosomes will remain as they are. No replication between the first and the second meiotic division. So it goes immediately into Prophase 2. So let's go on. And now we have the two cells, and they're going through Prophase 2. Again, the only difference between what we had as Telophase 1, so here Telophase 1, and Prophase 2, so let's label it like this, Prophase 2. The only difference really that happened is that, of course, the centrosome is going to divide, forming two centrioles, and each of them will start to migrate to the opposite side of the cell. However, the chromosomes also undergo a slight decondensation in telophase, in between telophase and prophase, and in prophase they undergo condensation once again. Now in meiosis 2, you're not going to get genetic recombination again in prophase 2. We already had that in prophase 1. In meiosis 2, we do not have that process. Now moving on from prophase 2, the next stage is, of course, going to be metaphase. So we're going to label this as Metaphase 2. And metaphase 2, again, the chromosomes line up in the center of the cell, called the cell plate or metaphase plate. And now the kinetochore structure has formed on the centromere, and they're going to be pulled apart by the microtubules to its part of the cell. Now, there is no tetrad formed in metaphase 2. As you can see, there's no tetrad because there's only 2 chromosomes. We do not have another chromosome to make up a tetrad. Next, what happens is we enter into anaphase. Now, anaphase, of course, the cytoplasm is going to elongate due to the microtubules pulling it apart from the cell plate. What's important to note is that the chromosome, the homologous pair as we had here, has split up into sister chromatids. So each one of these is a sister chromatid, and they're being pulled to its side of the cell. And we are still remaining with the 2 chromosomes on each side of the cell. However, they are now sister chromatids. They're no longer homologous pairs. And this is exactly what happens in the process of mitosis. When you go from metaphase to anaphase, mitosis is exactly the same. So meiosis 2 is very similar to mitosis. So let's label it anaphase, anaphase 2. And finally, anaphase 2, of course, goes into the final stage where we have 4 different cells, 4 different gametes, and this is telophase 2. So these would all be, here let's color it gametes, so either sperm or egg cells, and they would have chromosomes, the haploid number of chromosomes, and each of these, as you can see, so this one just has the pink. However, this one has pink and a little bit of that bluish because of the genetic recombination from Meiosis One. This one, all of it is blue, and here from the bluish to a pink segment as well. The chromosomes obviously go through decondensation. Now they're unraveled and they're into this loose formation. And now they're gametes. They're either sperm or egg cells. They're going to go and find the opposite sex gamete. And they're going to go through the process of fertilization to form a zygote. And the cycle would begin again. So this concludes our video on meiosis and chromosomes. I hope this cleared up a lot of the misconceptions about chromosomes and meiosis and the haploid and the diploid stage, because it all gets super confusing. But I am really hoping this video has helped. Now, in the next video, we're going to take a look at chromosome maps and what those mean and how we make them.